Alright, so picking up with the endomembrane system. Alright, so the endomembrane system is a series of interconnected membranes. Alright, not all of them are connected, but a series of membranes that are involved in ultimately the synthesis and secretion of proteins. Alright, so the endomembrane system consists of the nuclear envelope, alright, the endoplasmic reticulum, alright, specifically when you're talking about the ER, right, the rough ER. Alright, the Golgi apparatus or the Golgi body, okay, lysosomes, vacuoles, and plasma membrane. Now your nuclear envelope is interconnected with the rough ER. Alright, now the rough ER is not directly connected to the Golgi apparatus, alright, but these two things, alright, the nuclear envelope and the ER are interconnected membranes. Alright. There are a series of microtubules that are a component of the cellular cytoskeleton that are used for transport of vesicles, alright, to the Golgi body, alright, from the rough ER. And then there are microtubule vesicles that extend from the Golgi body to the cell membrane. Alright, so for trafficking of vesicles from the Golgi body to the cell membrane itself. Alright, so as I mentioned, the endomembrane system is either a continuous series of interconnected membranes or they're connected through vesicles that run along tracks of microtubules. Alright, so here you're being shown an image of the endomembrane system. Alright, so one of the things that's actually missing out of this is going to be your nuclear membrane. Alright, that nuclear membrane is going to be here. Alright. And it's going to be interconnected with the endoplasmic reticulum, specifically the rough ER. And so here you have your messenger RNA that's going to come out of your nucleus, and it's going to be translated into a protein on a ribosome. Alright, this is on the rough ER. That protein will enter the lumen or cavity space of the ER. It'll bleb off as a vesicle from the ER and being transported to the Golgi body. Alright, so the portion of the Golgi body that's closest to the rough ER is referred to as the cis face of the Golgi body, and the one that's closer to the cell membrane is the trans. Alright, so think about it this way. For something to be secreted and to be secreted to the cell membrane, it has to migrate across the cell, migrate across the Golgi body, so the opposite end of the Golgi body is trans, so across. Alright, so as the protein from the ER inside one of these little vesicles here makes its way to the Golgi body, alright, it goes to the Golgi body, it gets sorted, it gets tagged, and it gets directed to cell membrane, or it can get directed to a lysosome, or it can be a little storage, all right, vesicle. All right, so when you're talking about the endoplasmic reticulum, all right, you have rough ER and you have smooth ER. All right, so both of these components of the endoplasmic reticulum are interconnected with one another, as well with the nuclear envelope. All right, so your rough ER. Your rough ER is involved in basically synthesizing proteins that are going to be secreted. All right, that's one of its ultimate goals. Proteins are going to be secreted. All right, that's its job. The smooth ER. All right, it doesn't have any ribosomes that stud its surface. All right, so it's smooth. It's involved in basically detoxification of things like drugs and alcohol. All right, as well as storage of calcium ions. All right, so here what you're looking at is the rough ER. All right, so you have your messenger RNA transcript. All right, your messenger RNA transcript, all right, is going to interact with your ribosome, which again is involved in synthesizing proteins. All right, and as that protein is synthesized, it'll actually pass through a pore in the rough ER. Here, it enters a pore, enters the luminar cavity of the ER. Here it can be modified, where you can have sugar residues that get attached. Alright, so you get a glycoprotein, sugar protein. Alright. And now the membrane of the ER will bleb out and encase this protein, forming a secretory vesicle. Alright, so here, looking at smooth ER. Alright, so muscle tissue. Alright, so for instance, looking at cardiac muscle tissue, you have something called the sarcoplasmic reticulum. Basically, this is the smooth ER. Alright, so this is where you have storage of calcium ions. Alright, and these calcium ions are released, and when they're released, they aid in muscle contraction. Alright, so the smooth ER serves as kind of a surplus. Alright, a bulk stock of calcium ions that are stored until they're needed. Alright, so the Golgi Body, or Golgi Apparatus. Alright, think of this as the Postal Service of the cell. Alright, it's one big post office. Alright, so this is where you have your molecules that are synthesized, and they're transported to the Golgi Body, so specifically your proteins. Alright, and they get sent to the Golgi Body. When they're in the Golgi Body, they are going to be basically sorted, tagged, and delivered to specific locations. Alright, so basically you get a stamp and a forwarding address attached to a biological molecule, and it gets delivered to where it needs to go. Alright, so here you have your secretory vesicle that's coming from the rough ER. Alright, it's going to fuse with the membrane of the Golgi Body. Now, the contents of that vesicle are now in the lumen, or cavity, of the Golgi Body. And as that molecule makes its way through a series of interconnected membranes, and it moves from the cis side, alright, to the trans side, it's going to be modified and sorted. So then it's going to be directed to where it needs to go. Alright, so the trans side is the shipping side. Alright, so basically think of the cis side as the packages that are coming into the post office, and the trans side as the packages that are leaving the post office. Okay. Alright, so this is kind of giving you a three-dimensional image of your Golgi complex. Alright, so a series of interconnected membranes. Alright, so this particular image is showing you, alright, what's going on at the, alright, so here you have your cis side. Alright, you have your secretory vesicle here with your protein. Alright, it's going to fuse with the membrane of the Golgi Body, and the contents of that particular vesicle are going to be spilled into the lumen of the Golgi Body. Okay. Now, that protein will get a chemical tag attached to it. And this chemical tag is basically labeling it for a specific location. Alright, so some proteins are directed to the cytosol, alright, or to other organelles, or they're transported to the lysosome, or they're transported to the plasma membrane. Alright, and each tag directs that protein or group of proteins to a specific location in the cell. Alright, and these particular proteins will go from the trans side of the Golgi Body. They'll exit the membrane of the Golgi Body that encases them, wraps around them, forming a secretory vesicle that's transported to some other location in the cell. Alright, so your lysosome. Your lysosome is a product of the endomembrane system. It's a part of the endomembrane system, but it's also a product of it. Alright, so it all starts in the nucleus, goes from the nucleus to the rough ER, from the rough ER to the Golgi Body, to the Golgi Body to forming your lysosome. Alright, so your lysosomes contain digestive enzymes. Alright, so you have, and here we have a lysosome, and you're shown the enzymes that are contained. So you have nucleases that break down DNA and break down RNA. You have proteases that break down proteins. You have glycosidases that break down sugars. You have lipases that break down fats. You have phosphatases, sulfatases, phospholipases that break down other biological molecules. Alright, so it's one big, basically, digestive soup that's involved in degrading and breaking down biological molecules into their little minuscule parts. Alright, so lysosomes are involved in two processes, one of which is autophagy. Alright, so if you have an organelle that's getting old or it's damaged, here you have the mitochondria, for instance, and the cell will basically recycle that organelle, all right, the biological molecular components of it. All right, so that organelle will be wrapped around certain proteins that'll direct it to a lysosome. That lysosome will fuse with it, so you get this lysosome, phagosome type of vesicle that forms, phagolysosome, that will begin to digest your little mitochondria, and then all the small biological molecules and building blocks get recycled. Alright, so it functions as a recycling factory. Alright, and then for things like your white blood cells or something like an amoeba, alright, these guys use phagocytosis to engulf either foreign matter, like bacteria, or an amoeba engulfs bacteria as food. Alright, so here you get this phagosome that forms, which is basically a vesicle. When your cell wraps pseudopodia around your little bacterial cell here, all right, and it enters the cytoplasm. All right, your lysosome fuses with your phagosome, forming a phagolysosome, and then the constituents get broken down, and any small molecules are going to be recycled. Alright, so this is how your white blood cells destroy foreign invaders, and how an amoeba obtains nutrients from its environment. Alright, so here you're being shown a process of phagocytosis. Alright, so here in red, you have this red spherical structure is being basically phagocytized. You can see that it's going to be inside of a vesicle. Alright, so here you have your little bitty dye molecules being taken up by this, probably is a white blood cell, and it gets trapped in this little phagosome, and that phagosome is going to fuse eventually with some lysosome in the cell. Alright, but this dye is probably, it may be a small bacterium, it may be a food particle, this organism is picking up from the environment. Alright, so don't get vacuoles and vesicles confused. Okay. Alright, so vacuoles are really big vesicles. Alright, so you have secretory vesicles that are pretty small, and you have these large vesicles that are called vacuoles. Now, different cell types have different types of vacuoles. Alright, so your amoeba can have basically these little vesicles that form around food particles. And these would be referred to as food vacuoles. All right. You have your protist. All right. A lot of your animal-like protist, like you'll see in a minute, like a little paramecium, has a contractile vacuole. All right. So a contractile vacuole is involved in what we call osmoregulation. All right, so it's trying to regulate the amount of water inside the cell in relation to the outside environment. All right, so in this case, the contractile vacuole is involved in expelling excess water from the cell. All right, so recall that the animal cells, eukaryotic cells, for instance, like your little protist, they don't have a cell wall, right? So that means that they take up too much water, they pop, they burst. So they have to have some way to basically expel excess water. Right. And then you have plant cells that have central vacuoles. So the central vacuoles are going to store water. All right, they maintain the rigidity of the cell, and they also store other types of molecules. All right, so things like pigments. All right, specifically pigments that make up the colors of your flower petals. All right, you have storage of food in vacuoles. All right, so food vacuoles. And then you have the storage of excess water in your central vacuole. The central vacuole will also store poisons and toxins and noxious chemicals that will be used to deter predation. And here you have a little paramecium. All right, so your little paramecium, notice here you have this vacuole here. That's a contractile vacuole. All right, so this little paramecium is swimming around in pond water. All right, so that means that the internal constituents of the cell have a higher concentration of solutes. All right, so there's less water. All right, so water rushes in to the cell. All right, so to keep the cell from bursting, all right, this little contractile vacuole will swell with water, and then it'll push out excess water, all right, to maintain the right concentration of solutes and water in the cell. All right, so here it's filling, and here you can see that the contractile vacuole has decreased to its normal size. And what you can see are these little bitty tubes that come out, that radiate out. All right, these little tubes are going to lead out to the membrane of the cell so that they can basically expel water from the little bitty paramecium. All right, so here you're being shown an image of a paramecium as contractile vacuole. Now the contractile vacuole is going to be found in here. All right, so you're going to want to focus your attention to that part of the screen. All right, so here it begins to swell and then pushes water out. So you can see in the circle where that vacuole is swelling and then dispelling excess water. It's expanding again. and it displaces the water. Alright, so this is giving you an overview of the endomembrane system. Alright, so your endomembrane system is all about secretion of proteins. Alright, so it all starts with the messenger RNA coming out of the nucleus. That messenger RNA is going to be translated into a protein along the ribosomes that are studded on the surface of the rough ER. That protein is then going to be transported through a secretory vesicle to the Golgi body. Here, your protein will be modified and tagged and directed to where it needs to go. So it could become a component of the lysosome. Alright. It can become a vesicle that sits out in the cytosol, alright, because it has proteins that are used in the cytosol itself. Or these proteins will be expelled from the cell through exocytosis. Or the proteins become embedded in the membrane of the cell. Alright, so here, this video is showing you these little vesicles. They're actually being trafficked. They're being transported, that's a better word to use, transported to the Golgi body. Alright, so these vesicles aren't just randomly moving around the cell. Alright, they're actually being physically transported through basically like a little bitty, you want to think about it, little UPS truck, little mail truck. And this little mail truck is running along a little road or track, if you will, of microtubules. And it's being directed from the ER to the Golgi body. Alright, so all these vesicles are being directed towards the Golgi body. Alright, so you can see these vesicles are moving along a track. Alright, little rail system. Alright, to the Golgi body. Here, you're looking at the Golgi body as it's secreting proteins, as the little vesicles are plopping off. Alright, so things are plopping off the Golgi body. Little secretory vesicles coming off of it. Alright, so next we're going to look at your mitochondria and your chloroplast. Alright, so mitochondria are involved in ATP synthesis, and your chloroplast is involved in photosynthesis. Alright, so making things like glucose and sucrose. Alright, so there's this theory called the endosymbiotic theory. Alright, and it kind of explains the origin of these two particular energy processing organelles. Alright, so there's evidence that supports the endosymbiotic theory. Alright, so one is that your mitochondria and your chloroplast are enveloped by a double membrane. Alright, so they have an inner membrane that is similar to a bacterial membrane. Alright, and then they have an outer membrane as well. which would be the product of basically the engulfment of a host cell. Because a host cell membrane, as it engulfs a specimen, all right, a portion of the membrane of the host cell becomes that vesicle. All right, so that would be the outer membrane. All right, your mitochondria and chloroplasts, they have 3 ribosomes. All right. And they have their own circular double-stranded DNA. All right, so mitochondrial DNA, chloroplast DNA is completely separate from nuclear DNA. All right, so mitochondrial and chloroplast DNA encode for specific proteins that are involved in energy processing in the mitochondria and chloroplasts, respectively. All right, so it increases the efficiency of these processes. You don't have to rely on the nucleus to carry out transcription and translation of these particular proteins. All right, it can all occur in-house in the mitochondria. And in your mitochondria and chloroplast, they replicate separately from the host cell. Also, your mitochondria and your bacterial cells and your chloroplast are all similar in size. All right, so the idea here is that you have this primitive ancestral eukaryotic cell. All right, it's got a membrane-bound nucleus. All right, it's got an endoplasmic reticulum, and it engulfs a oxygen-using non-photosynthetic bacterium. All right, this little guy here. And this bacterium is alive, it can live on its own, but it's engulfed by this ancestral eukaryotic cell. And over time, this little bacterial cell that can use oxygen to produce ATP, over time loses its ability to function on its own. All right, it loses its own identity, if you will. All right, and so it becomes an organelle. All right, that we call the mitochondria. All right, it still performs its job. It's just no longer a independent entity in a host cell. All right. Then comes along this little non-photosynthetic eukaryotic cell that engulfs a photosynthetic prokaryote. This photosynthetic prokaryote, all right, is able to utilize sunlight to produce food. That's beneficial for the host cell. All right, but over time, that little photosynthetic prokaryote again undergoes this process of losing its identity. It degenerates into a organelle. It's no longer a cell, but it still is able to carry out its basic functions of photosynthesis. So you end up with your eukaryotic, primitive plant and animal cells. Alright, so your mitochondria. Your mitochondria are going to be found in pretty much all eukaryotic cells. Alright, so both plant and animal cells. Alright, keep that in mind. Alright. Now, we're going to talk about the structure of the mitochondria. Alright, so your mitochondria has an outer membrane. Alright. It has an inner membrane, and this inner membrane is actually folded in on itself All right, these invaginations are referred to as cristae. All right, the cristae basically increase the surface area of the inner membrane. All right, now this is important. All right, because in the inner membrane are the proteins that are a component of the electron transport chain that's used in making ATP. All right, so between your outer membrane and your inner membrane, you actually have this void. All right, this lumen, this cavity, is referred to as the intermembrane space. And then inside the lumen of the mitochondria, inside the membrane, all right, you have this fluid-filled compartment that we call the matrix. In the matrix, this is where you find the ribosome, and this is where you find your mitochondrial circular DNA. Now, looking at your chloroplast, all right, so your chloroplast, all right, it also has an outer membrane, has an inner membrane, and it has this intermembrane space. All right, so inside of the chloroplast, all right, you have this fluid-filled space inside of it that we call the stroma. All right, in the stroma, you have your ribosomes. All right, you have your circular double-stranded DNA. All right, your chloroplast DNA. And then you have these stacks of discs. Now, these stacks of discs are what we refer to as collectively granum, singular, or many grana. And these stacks of discs, the discs are what we refer to as thylakoids. Now, the thylakoids also have their own internal lumen compartment. All right, there's a cavity inside of the thylakoids. All right, they're hollow on the inside. Well, not hollow, but I mean, there's a fluid-filled compartment inside, but I mean, they have a space, interior space inside the thylakoid. All right, so think of a granum or grana as a stack of coins, and each coin represents a thylakoid. All right, so later on we're going to talk about photosynthesis. All right, but for all intents and purposes, kind of give you a preview. All right, while we're talking about these structures. All right, so your thylakoid is involved in what we call the light reaction of photosynthesis. And your stroma is what's referred to as the Calvin cycle, or this archaic term called the dark reaction. All right, that's no longer really an appropriate term. All right, we use the Calvin cycle as the appropriate process that occurs. And these two together make up the process of photosynthesis. All right, so one of the other organelles that we have yet to talk about is the peroxisome. All right, so your peroxisome basically contains enzymes that break down reactive oxygen species. All right, things like hydrogen peroxide, which are a byproduct of aerobic cellular respiration. All right, so it's a natural byproduct, but the cell has to get rid of it. All right, because reactive oxygen species can target proteins, DNA, and lipids and damage them. All right, so that's not very beneficial for a cell. All right, so the cell has to be able to break down these toxic byproducts. All right, so it produces an enzyme called catalase. And this catalase is inside of your peroxisome, and it can break down the hydrogen peroxide and convert it into water and oxygen gas. Now, your peroxisomes are also involved in the beta oxidation, or the breakdown of your fatty acids. All right, so thinking about all these different types of cells. All right. Now, one of the beautiful things about your eukaryotic cells is that you have this process of various membrane-bound organelles and structures. And these membrane-bound structures or organelles allow for specificity and efficiency of carrying out very particular biological processes. Alright, so your mitochondria carries out the production of ATP, your chloroplasts carry out the process of photosynthesis, your rough ER carries out the process of aiding in the synthesis of secreted proteins, your lysosomes are involved in digestion. Alright, so you have this segregation of functions to different parts of the cell. Alright, so it speeds up the efficiency of the cell. Alright, so everything's micromanaged. Okay? So it can be more easily regulated by the cell too. Alright, now, each cell type is going to have its own proportion of organelles. Alright, so all of your animal eukaryotic cells have all of the, for all intents and purposes, is going to contain all of the particular organelles you would find in a normal animal cell. Alright, there's going to be exceptions. Alright, but in this case, if you're looking at an animal cell and you're looking at a skin cell versus a liver cell versus a heart muscle cell, these are three different types of cells that may have certain organelles in higher proportion than others because they all differ in function. Alright, so here you're looking at pancreatic cells. Alright, so pancreatic cells are involved in synthesizing, in this case, insulin. Alright, and insulin is actually secreted by the pancreas. Alright, so that means that the protein insulin secreted by pancreatic cells, that means that it's going to have a higher proportion of a particular group of organelles that are involved in secretion of proteins. So it has a higher proportion of rough ER and probably Golgi Body. Right. Your plant cell, alright, is involved in photosynthesis. Now this is a plant leaf cell. Alright, and leaves are is basically the organ of the plant that carries out light absorption. Alright, so they're going to have a higher proportion of chloroplast. All right, because they're the ones that are absorbing light energy used in making ATP and sugars. Now, animal testes. Animal testes are involved in exporting lipid-soluble signals. Things like, for instance, the hormone testosterone. Alright, so testosterone is a lipid. You know that it is a steroid. Alright, and it's basically going to be exported from the testes. And so since the testes is synthesizing this particular steroid, alright, one thing I haven't brought up is the fact that your smooth ER is involved in lipid synthesis. Okay, so lipids such as steroids, your for instance your phospholipids, all right, that make up the membrane of the cell, those are also synthesized by the smooth ER. Okay, and here, all right, you have brown fat cells. All right, so you have white adipose tissue, which stores basically triglycerides or fat molecules. Your brown fat cells are found in a higher proportion in your hibernating animals, things like bears, for instance. And so think about it. Your hibernating animals, they bulk up on food throughout the year. And then during the wintertime, they kind of hunker down and kind of keep themselves out of the elements. But how do they keep warm? Alright, well, they have brown fat cells. And brown fat cells are involved in releasing heat instead of ATP. Alright, and this also involves mitochondria. Alright, so the brown fat cells have a ton of mitochondria. Alright, so they can produce a lot of heat.